What are some methods of asexual reproduction?
Budding, fission, and fragmentation are methods of asexual reproduction. In budding, a new individual begins as an outgrowth, or bud, of the parent. Eventually, the bud detaches from the parent and develops into a new individual. Budding is common among sponges and coelenterates such as hydras and anemones. Fission is the division of one individual into two or more individuals of almost equal size. Each new individual develops into a mature adult. Some corals reproduce by dividing longitudinally into two smaller but complete individuals. Fragmentation is the breaking of the parent into several pieces. It is accompanied by regeneration when each piece develops into a mature individual. Sea stars are well known for reproducing by fragmentation and regeneration.
What is the difference between white rot fungi and brown rot fungi?
White rot fungi, found in the wood of deciduous trees, first attack the lignin of wood. Once the lignin is digested, the fungi destroy cellulose and other major parts of cells. The partially decayed wood with residual cellulose is off-white in color. Hence the name white rot fungi. Brown rot fungi, found in conifers. Damage the cellulose first but do very little, if any, damage to the lignin. The name brown rot fungi came about because infected wood becomes dark reddish brown to golden in color.
Which woods are recommended for construction when the wood will be constantly exposed to moisture?
Decay-resistant woods such as redwood and cedar or wood treated with preservatives are recommended for use in construction when the wood will be exposed to moisture. The most effective and least toxic preservative is chromated copper arsenate, CCA. Lumber treated with CCA has a characteristic light green stain.
What is the dikaryotic phase of fungal life cycles?
The dikaryotic phase of the fungal life cycle is unique. During this unusual phase, which is common in many species of fungi, cells contain two distinct nuclei. These two nuclei divide simultaneously as the mycelium grows. Growth continues until fusion occurs during karyogamy.
How many species of fungi have been identified?
Scientists have identified between 70,000 and 80,000 species of fungi, almost 2,000 new species are discovered each year. Some mycologists estimate that there are about 1.5 million species worldwide, placing the fungi second only to insects in the number of different species.
Where does rain come from?
Solar energy drives winds that evaporate water from the surface of the oceans. The water vapor cools as it rises and then falls to the ground as rain. Snow, or some other form of precipitation. Rain is part of the hydrologic cycle, which describes dynamic changes in the aquatic environment.
What is thermodynamics?
Thermodynamics is the study of the relationships between energy and the activity of a cell. The laws of thermodynamics govern the way in which cells transform chemical compounds. The first law of thermodynamics states that the total energy of a system and its surroundings will always remain constant. The second law of thermodynamics states that systems, such as a cell, tend to become disordered. Disorder usually arises in the form of heat. Entropy is the term that refers to the disorder that tends to disrupt cells, or even the universe.
Who decides which way a troop of monkeys will travel?
Among savanna and gelada baboons, females form the stable social structure of the troops while males transfer between troops. Observations of gelada troops have shown that it is the females who determine the direction the group will travel during the daily foraging trips.
What is bioterrorism?
Bioterrorism is the use of biological substances or toxins with the goal of causing harm to humans. Biotechnology can be used to manufacture biological weapons such as large amounts of anthrax spores. However, biotechnology can also be used positively to identify bioweapons. A new faster method of PCR, called continuous flow PCR, uses a biochip and requires only nanoliter amounts of DNA to detect a bioweapon.
How do fossils form?
Fossils form rarely, since an organism is usually consumed totally or scattered by scavengers after death. If the structures remain intact, fossils can be preserved in amber, hardened tree sap. Siberian permafrost, dry caves, or rock. Rock fossils are the most common. In order to form a rock fossil, three things must happen: 1. The organism must be buried in sediment, 2. The hard structures must mineralize, and 3. The sediment surrounding the fossil must harden and become rock. Many rock fossils either erode before they can be discovered, or they remain in places inaccessible to scientists.
What is scientific creationism?
Scientific creationism is an attempt to promote the teaching of creation theory in schools. By designating creationist theory scientific, proponents hope to gain equal time with evolutionary theory in school curricula. Creationist theory proposes that species observed today are the result of intelligent design or special creation rather than the result of the effects of natural selection.
What cell structures are unique to animal cells?
Lysosomes and centrioles are found only in animal cells.
What are the non-chilomates?
The non-chilomates are animals that lack a body cavity. A chylome, and include the sponges, jellyfishes, and simple worms.
What are the advantages of gel electrophoresis?
In gel electrophoresis, the molecules are forced across a span of gel, usually agarose. Gels are easy to modify and have an excellent power of separation. The frictional force of the gel material acts as a molecular sieve, separating the molecules based on their size.
Are any other animals able to drink seawater?
Birds and reptiles that live near the sea are also able to drink seawater. These animals have nasal salt glands near their eyes through which they excrete the excess quantities of salt solution.
Is DNA always copied exactly?
Considering how many cells there are in the human body and how often it occurs, DNA replication is fairly accurate. Spontaneous damage to DNA is low, occurring at the rate of 1 to 100 mutations per 10 billion cells in bacteria. The rate for eukaryotic genes is higher, about 1 to 10 mutations per million gametes. The rate of mutation can vary according to different genes in different organisms.
What is anthropomorphism?
Anthropomorphism is the attribution of human characteristics and feelings to non-humans. An example of this is Bambi, a story written by Felix Salten, 1869 to 1945, in 1923. The inspiration for the story came from the wildlife he saw while vacationing in the Alps. When the story was eventually made into a Disney movie, Bambi had become a talking animal, complete with human feelings and emotions. Anthropomorphism can obscure the true motivation for an animal's behavior.
How wide is the lateral spread of the root system of trees?
The lateral spread of a tree's root system is 4 to 7 times greater than the spread of the crown of the tree.
What is the Calvin cycle?
Photosynthesis creates reactions that are both light-dependent and light-independent. The Calvin cycle is part of the reaction that is light-independent and occurs in the chloroplast. This cycle is crucial to the capture of carbon dioxide, leading to the formation of sugar, C6H12O6. It is named in honor of Melvin Calvin, 1911 to 1997, who received the 1961 Nobel Prize in Chemistry for unraveling the process of glucose biosynthesis.
Who was Nico Tinbergen?
Nico Tinbergen, 1907 to 1988, along with Konrad Lorenz, 1903 to 1989, and Carl von Frisch, 1886 to 1982. Laid the four cornerstones of ethological study: causation, development, evolution, and function of behavior. Tinbergen's earliest studies were of the aggressive threat displays of adult male three-spined stickleback fish, Gasterosteus aculeatus. Tinbergen found that by presenting the male with stickleback models, he was able to elicit an aggressive response. He began his career in animal behavior as a child by studying the mating and nesting behavior of sticklebacks in his backyard pond. He attended the University of Leiden, where he did his dissertation research on digger wasps. His thesis was only 36 pages long and was barely accepted by the faculty. In 1936 he met Lorenz at a symposium, and they became lifelong friends and colleagues. During World War II, Tinbergen was imprisoned in a German hostage camp and Lorenz in a Russian camp. When they were reunited after the war, both resumed their studies on animal behavior.
Why were such dangerous chemicals as DDT, PCBs, and CFCs released into the environment?
Dichlorodiphenyl trichloroethene, DDT, polychlorinated biphenyls. PCBs, and chlorofluorocarbons, CFCs, were once widely used. Although DDT was synthesized as early as 1874 by Othmar Zeidler, it was the Swiss chemist Paul Müller, 1899 to 1965, who recognized its insecticidal properties in 1939. He was awarded the 1948 Nobel Prize in Physiology or Medicine for his development of DDT. Unlike the arsenic-based compounds then in use, DDT was effective in killing insects and seemed not to harm plants and animals. In the following 20 years it proved to be effective in controlling disease-carrying insects. Mosquitoes that carry malaria and yellow fever, and lice that carry typhus, and in killing many plant crop destroyers. Publication of Rachel Carson's book Silent Spring in 1962 alerted scientists to the detrimental effects of DDT. Increasingly DDT-resistant insect species and the accumulative hazardous effects of DDT on plant and animal life cycles led to its disuse in many countries during the 1970s. In fact, DDT and PCBs have been added to the list of chemicals known as estrogenic compounds, that is, synthetic substances in the environment that cause the mammalian body to respond as if to estrogen. A group of chemicals with the same general chemical structure and physical properties as DDT are known as the polychlorinated biphenyls or PCBs. Because of their physical properties, non-flammability, chemical stability, high boiling point, and electrical insulating properties, PCBs can be used in a variety of applications. Formerly, many products contained these compounds. From electrical circuitry to the dyes and pigments used in paint, to carbonless copy paper, all were manufactured with PCBs. Before production was ceased in 1977, the United States produced about 1.5 billion pounds, 6.8 billion kilograms, of PCBs. Chlorofluorocarbons, CFCs, are commonly used as aerosol sprays, refrigerants, solvents, and foam-blowing agents. They are in and of themselves non-toxic and non-flammable molecules containing chlorine, fluorine, and carbon. However, they are thought to have a deleterious effect on ozone concentrations in the atmosphere.
What are the dangers involved with transgenic animal transplants?
One of the major risks of xenotransplantation is the risk of transplanting animal viruses along with the transfer cells or organs. Since the patient is already immunosuppressed, the patient could die from the viral infection, or the virus could be spread to the general population.
What are the largest and smallest aerial spider webs?
The largest aerial webs are spun by the tropical orb weavers of the genus Nephila, which produce webs that measure up to 18.9 feet 6 meters in circumference. The smallest webs are produced by the species Glyphosis cottoni. Their webs cover an area of about 0.75 sq in, 4.84 sq/cm.
What happens if a cell's DNA is mutated?
DNA contains the code for building everything within the cell. If the DNA code is changed, or mutated, the cell will build things differently. Mutations can change the way that a cell produces substances, such as proteins and carbohydrates, builds organelles, or responds to messages. These changes can enhance a cell's ability to survive. Although it is more likely that drastic changes will result in a decrease of efficiency or even the death of the cell.
What important scientific discoveries were made using X-ray diffraction?
In 1951 the protein A helix was discovered by Linus Pauling, 1901 to 1994. X-ray diffraction was used to reveal the double helix structure of DNA in 1953 by Maurice Wilkins. 1916, Francis Crick, 1916 to 2004, Rosalind Franklin, 1920 to 1958, and James Watson, 1928. Dorothy Mary Crowfoot Hodgkin, 1901 to 1994. Used the technique to determine the structure of vitamin B12 in 1956.
How is it possible to measure the pH of a solution?
An easy way to measure the pH of a solution is with pH paper. This paper is treated with a chemical indicator that changes colors. Depending on the concentration of H+, hydrogen ions, in the solution.
Why do some biologists consider the insects the most successful group of animals?
With more than 1 million described species, and perhaps millions more not yet identified. Class Insecta is the most successful group of animals on Earth in terms of diversity. Geographic distribution, number of species, and number of individuals. More species of insects have been identified than of all other groups of animals combined. What insects lack in size, they make up for in sheer numbers. If we could weigh all the insects in the world, their weight would exceed that of all the remaining terrestrial animals. About 200 million insects are alive at any one time for each human.
HAT is the historical evidence of a genetic disease?
One example of historical evidence of a genetic disease is found in Egyptian art of 3,500 years ago that shows Akhenaten, pharaoh of Egypt during the 18th dynasty. Akhenaten is depicted as a man with a sharp chin, almond-shaped eyes, full lips, long arms and fingers, and enlarged breasts. One early statue shows the king in the nude and without genitalia. These features have intrigued archaeologists, and two theories have been proposed for his odd appearance. One is that Akhenaten suffered from either Froelich syndrome or Marfan syndrome. Froelich syndrome is an endocrine disorder found most commonly in men. This disorder affects development of secondary sex characteristics, which fit some of the characteristics associated with Akhenaten's appearance. However, Akhenaten, although he had some feminine characteristics, was known to have fathered at least 6 children. And he was not retarded, another characteristic of Froelich syndrome. The more likely possibility is Marfan syndrome, which results in patients with long fingers. Skeletal abnormalities, but normal intelligence and fertility. Also, evidence suggests that the rest of his family apparently showed some of these traits, suggesting that he had a genetic disorder.
What is a prion?
Prions are abnormal forms of natural proteins. Current research indicates that a prion is composed of about 250 amino acids. Despite extensive and continuing investigations, no nucleic acid component has been found. Like viruses, prions are infectious agents.
Do animals ever help each other?
Populations of organisms within an environment may engage in a variety of relationships with each other. For example, in a relationship known as mutualism, each species provides a benefit to the other. Mutualism can occur between 2 animal species, like the relationship between large coral reef fishes and the smaller species like the wrasses that swim into their mouths and eat the parasites that may have taken up residence there. Mutualistic relationships also allow bean plants to grow better because they exchange nutrients with rhizobium fungi living on their roots.
What is the ENCODE project?
The ENCODE, Encyclopedia of DNA Elements, project refers to a long-term project sponsored by the National Human Genome Research Institute to identify and locate all genes, including protein coding and non-protein coding ones, and other functional elements within the human genome.
How can speciation occur?
If a population becomes reproductively isolated, then individuals within the population will no longer exchange genetic material with the rest of the species. At that point, environmental factors, i.e., natural selection, will work on the genetic variation within that population until it has become a new species.
What is amber?
Amber is the fossilized resin of trees. Resin is the sticky material often seen oozing from the trunk of a pine tree. Resin hardens as it dries and is the source of both turpentine and rosin. Resin comes primarily from kauri pine forests. The translucent material emerges from trees and forms lumps that appear deep orange or yellow in color. These lumps may weigh up to 99 pounds 45 kilograms. The two major deposits of amber are in the Baltic region and the Dominican Republic. Amber has also been found in the central Appalachian region of the United States. Prehistoric insects have been so remarkably preserved in amber that the pieces still contain the insect's intact DNA. Amber is the only jewel of plant origin.
What is the purpose of a mordant?
A mordant is a chemical added to the solution used to stain a specimen in order to intensify the stain. Two major functions of a mordant are to increase the affinity of a stain for a biological specimen, and to coat a structure to make it thicker and easier to see upon observation under the microscope.
What is a better source of energy, fat or sugar?
Fats contain 9 kcal/g, while carbohydrates average about 4 kcal/g. Fats are useful for storing energy, but sugars are easily broken down during metabolism. Nutritionists and diet gurus are debating which source of energy is better. The absolute answer lies in individual situations. For example, babies require fat in their diets for both energy 24 and to build a healthy nervous system. Middle-aged adults quite often take in too much fat for their metabolic requirements. As one ages, average energy needs decrease by 5% per decade.
Are all large fungi shaped like mushrooms?
The fruiting bodies of fungi come in a seemingly endless array of forms and colors. Many are variations on the familiar stalk and cap pattern of the common mushrooms sold in stores. Although some have minute spore-bearing pores instead of gills on the undersides of their caps. Many fungi do not resemble mushrooms at all. Puffballs are solid, fleshy spheres. Bird's nest fungi form little cups containing eggs packed with spores. One kind of fungus looks like a head of cauliflower, and others resemble upright, branching clumps of coral. Some protrude from tree trunks like shelves, while others look like glistening blobs of jelly.
Why is DNA such a stable molecule?
DNA is a stable molecule due to the stacking interaction between the adjacent base pairs in the helix and the hydrogen bonding between bases. This stability has allowed researchers to harvest DNA from extinct species.
What is a chemical bond?
A chemical bond is an attraction between the electrons present in the outermost energy level or shell of a particular atom. This outermost energy level is known as the valence shell. Atoms with an unfilled outer shell are less stable and tend to share, accept, or donate electrons. When this happens, a chemical bond is formed. In living systems, chemical reactions, supervised by enzymes, link atoms together to form molecules.
Can humans make all the lipids they need?
No. There are two types of fatty acids that we need but can't make: linoleic acid, an omega-6 fatty acid, and linolenic acid, which is also known as an omega-3 fatty acid. These acids are used to maintain cell membranes and to build hormone-like messengers known as eicosanoids. Eicosanoids are 20-carbon fatty acids. Examples include leukotrienes and prostaglandins. Good sources of omega-3 and omega-6 lipids are nuts, grains, and both fats and oils from vegetables or fish.
Do viruses contain both DNA and RNA?
Viruses have either DNA or RNA as their genomic material, whereas cells including bacteria have both.
What is genetic engineering?
Genetic engineering, also popularly known as molecular cloning or gene cloning, is the artificial recombination of nucleic acid molecules in a test tube, their insertion into a virus, bacterial plasmid, or other vector system, and the subsequent incorporation of the chimeric molecules into a host organism in which they are capable of continued propagation. The construction of such molecules has also been termed gene manipulation because it usually involves the production of novel genetic combinations by biochemical means. Genetic engineering techniques include cell fusion and the use of recombinant DNA or gene splicing. In cell fusion, the tough outer membranes of sperm and egg cells are removed by enzymes. And then the fragile cells are mixed and combined with the aid of chemicals or viruses. The result may be the creation of a new life form from two species, a chimera. Recombinant DNA techniques transfer a specific genetic activity from one organism to the next through the use of bacterial plasmids. Small circular pieces of DNA lying outside the main bacterial chromosome. And enzymes, such as restriction endonucleases, which cut the DNA strands, reverse transcriptase, which makes a DNA strand from an RNA strand, DNA ligase, which joins DNA strands together. antic polymerase, which can make a double-strand DNA molecule from a single-strand primer molecule. The recombinant DNA process begins with the isolation and fragmentation of suitable DNA strands. After these fragments are combined with vectors, they are carried into bacterial cells, where the DNA fragments are spliced onto plasmid DNA that has been opened up. These hybrid plasmids are then mixed with host cells to form transformed cells. Since only some of the transformed cells will exhibit the desired characteristic or gene activity, the transformed cells are separated and grown individually in cultures. This methodology has been successful in producing large quantities of hormones, such as insulin, for the biotechnology industry. However, it is more difficult to transform animal and plant cells. Yet the technique exists to make plants resistant to diseases and to make animals grow larger. Because genetic engineering interferes with the processes of heredity and can alter the genetic structure of our own species, there is much concern over the ethical ramifications of such power, as well as the possible health and ecological consequences of the creation of these bacterial forms. Some applications of genetic engineering in various fields are agriculture, crops having larger yields, disease and drought resistancy, bacterial sprays to prevent crop damage from freezing temperatures, and livestock improvement through changes in animal traits. Industry, use of bacteria to convert old newspaper and wood chips into sugar. Oil and toxin-absorbing bacteria for oil spill or toxic waste cleanups, and yeasts to accelerate wine fermentation. Medicine, alteration of human genes to eliminate disease, experimental stage. Faster and more economical production of vital human substances to alleviate deficiency and disease symptoms, but not to cure them. Substances include insulin, interferon, cancer therapy, vitamins, human growth hormone ADA, antibodies, vaccines, and antibiotics. Research, modification of gene structure in medical research, especially cancer research. Food processing, rennin, enzyme, in cheese aging.
What emotions do animals have?
Many pet owners say that they know when their animal is happy or sad. And now there is evidence to show that animals do exhibit emotion. Researchers have found that emotions are accompanied by biochemical changes in the brain that can be measured. When scientists examine the physiological changes found in humans that correlate with certain emotional states, e.g., anger, fear, lust, they find that these changes can also be observed in certain animal species. A study of stress among African baboons showed that social behavior, personality, and rank within the troop can influence the levels of stress hormones. There is increasing evidence that birds, reptiles, and fishes also experience some form of emotions. Although the idea of animals feeling emotions raises skepticism, students of animal behavior agree that many creatures experience fear. which is largely instinctive and, in effect, is pre-wired into the brain. Field observations have recorded expressions that correlate with pleasure, play, grief, and depression. Jane Goodall, watching the reaction of a young chimp after the death of his mother, maintains that the animal died of grief. Even with this evidence, it is impossible to truly know how another organism feels.
What is the law of segregation?
This principle, which Mendel discovered, predicts the transmission of a single trait. When a gamete is produced, the two copies of a particular gene, alleles, separate during its formation. For example, if a pea plant has the genetic makeup of RR, where R is round and R is wrinkled, then half the gametes will contain R and the other half R. The law of segregation comes into play during meiosis when an organism decreases its chromosome number by half when forming egg or sperm.
What groups of reptiles are living today?
The three orders of reptiles that are alive today are, 1. Chelonia, which includes turtles, terrapins, and tortoises, 2. Squamata, which includes lizards and snakes, and 3. Crocodilia, which includes crocodiles and alligators.
What does it mean when a lake is brown or blue?
When a lake is brown, it usually indicates that eutrophication is occurring. This process refers to premature aging of a lake, when nutrients are added to the water, usually due to runoff, which may be either agricultural or industrial in origin. Due to this rich supply of nutrients, blue-green algae begin to take over the green algae in the lake, and food webs within the lake are disturbed, leading to an eventual loss of fish. When a lake is blue, this usually means that the lake has been damaged by acid precipitation. The gradual drop in pH caused by exposure to acid rain causes disruption of the food webs, eventually killing most organisms. The end result is clear water, which reflects the low productivity of the lake.
How do continuous feeders differ from discontinuous feeders?
Continuous feeders, also known as filter feeders, are aquatic animals that constantly feed by having water filled with food particles, e.g., small plankton or fishes, entering through the mouth. Continuous feeders do not need a storage area, such as a stomach, for food. Discontinuous feeders must hunt for food on a regular basis. They need a storage area to house food until it is digested.
What is a red tide?
A red tide occurs when there is a population explosion among toxic red dinoflagellates. Members of the genera Gymnotodium and Goniolax, both protistans that have an unusual cellular plate or armor. The population explosion, referred to as a bloom, may tint the water orange, red, or brown and can be toxic to shellfish, birds, and humans who eat red tide contaminated food.
Can cells be seen without a microscope?
Yes, but not many. Most cells are far smaller than the period at the end of the sentence. Bird and frog egg cells may be somewhat larger and can in fact be observed by the unaided eye.
What is meant by the phrase pecking order?
Pecking order refers to the dominance hierarchy, i.e., relative ranking, of animals within the same species. It has been adapted in business to refer to one's position within the company.
Who was awarded 2 Nobel Prizes for DNA sequencing?
Frederick Sanger, 1918, won the Nobel Prize in Chemistry in 1958 for his work on the structure of proteins, especially insulin, and shared the Nobel Prize in Chemistry in 1980, along with Walter Gilbert. 1932, for contributions in determining the nitrogen base sequences of DNA. This method was later referred to as the Sanger sequencing method for reading DNA.
What has been the impact of zebra mussels on North American waterways?
Zebra mussels, Dreissena polymorpha, are black and white striped bivalve mollusks. They are hard-shelled species that adhere to hard surfaces with byssal threads. They were probably introduced to North America in 1985 or 1986 via discharge of a foreign ship's ballast water into Lake St. Clair. They have spread throughout the Great Lakes, the Mississippi River, and as far east as the Hudson River. High densities of zebra mussels have been found in the intakes, pipes, and heat exchangers of waterways throughout the world. They can clog the water intakes of power plants, industrial sites, and public drinking water systems. Foul boat hulls and engine cooling water systems, and disrupt aquatic ecosystems. Water processing facilities must be cleaned manually to rid the systems of the mussels. Zebra mussels are a threat to surface water resources because they reproduce quickly, have free-swimming larvae and rapid growth, lack competitors for space or food, and have no predators.
What is the light source for a spectrophotometer?
Most commonly, a spectrophotometer uses white light as its light source. White light is a combination of all visible wavelengths. Ultraviolet and infrared wavelengths may also be used.
Do all animals have blood?
Some invertebrates, such as flatworms and cnidarians, lack a circulatory system that contains blood. These animals possess a clear, watery tissue that contains some phagocytic cells, a little protein, and a mixture of salts similar to seawater. Invertebrates with an open circulatory system have a fluid that is more complex. It is usually referred to as hemolymph, from the Greek term haimo, meaning blood, and the Latin term lympha, meaning water. Invertebrates with a closed circulatory system have blood that is contained within blood vessels. Vertebrates have blood composed of plasma and formed elements.
How deep does the root system penetrate the soil?
The depth to which the root system penetrates the soil is dependent on moisture, temperature, the composition of the soil, and specific plant. Most of the roots actively absorbing water and minerals. The feeder roots are found in the upper 3 feet 1 meter of the soil. The feeder roots of many trees are mainly in the upper 6 in 15 centimeters of the soil, the part of the soil richest in organic matter.
What are the major types of connective tissue and their function?
The major types of connective tissue are 1. Loose connective tissue, 2. Adipose tissue, 3. Blood. 4. Collagen, sometimes called fibrous or dense connective tissue, 5. Cartilage, and 6. Bone. Loose connective tissue is a mass of widely scattered cells whose matrix is a loose weave of fibers. Many of the fibers are strong protein fibers called collagen. Loose connective tissue is found beneath the skin and between organs. It is a binding and packing material whose main purpose is to provide support to hold other tissues and organs in place. Adipose tissue consists of adipose cells in loose connective tissue. Each adipose cell stores a large droplet of fat that swells when fat is stored and shrinks when fat is used to provide energy. Adipose tissue pads and insulates the animal body. Blood is a loose connective tissue whose matrix is a liquid called plasma. Blood consists of red blood cells, erythrocytes, white blood cells, leukocytes, and thrombocytes, or platelets, which are pieces of bone marrow cell. Plasma also contains water, salts, sugars, lipids, and amino acids. Blood is approximately 55% plasma and 45% formed elements. Blood transports substances from one part of the body to another, and plays an important role in the immune system of the animal. Collagen, from the Greek kolla, meaning glue, and genos, meaning descent, is a dense connective tissue, also known as fibrous connective tissue. It has a matrix of densely packed collagen fibers. There are two types of collagen, regular and irregular. The collagen fibers of regular dense connective tissue are lined up in parallel. Tendons, which bind muscle to bone, and ligaments, which join bones together, are examples of dense regular connective tissue. The strong covering of various organs, such as kidneys and muscle, is dense irregular connective tissue. Cartilage, from the Latin cartilago, meaning gristle, is a connective tissue with an abundant number of collagen fibers in a rubbery matrix. It is both strong and flexible. Cartilage provides support and cushioning. It is found between the discs of the vertebrae in the spine, surrounding the ends of joints such as knees, and in the nose and ears. Bone is a rigid connective tissue that has a matrix of collagen fibers embedded in calcium salts. It is the hardest tissue in the body, although it is not brittle. Most of the skeletal system is comprised of bone, which provides support for muscle attachment and protects the internal organs.
Why do animals in a zoo pace?
Pacing is an indication of lack of stimulation. A recent doctoral study found that larger animals, which have a larger home range in the wild, are particularly prone to pacing in captivity, a behavior known as cage stereotyping. Most reputable zoos attempt to assuage pacing by providing what is known as animal enrichment. Enrichment may include constantly changing objects, odors, and sounds to mimic the types of stimulations that would be found in the animal's natural habitat. As an example, in order to keep life interesting for poison dart frogs from South America, keepers will hide their crickets inside a coconut with holes drilled in it. The frogs then have to seek out the food inside the chirping coconut.
How do bacterial and fungal spores differ?
The main purpose of bacterial spores known as endospores is to protect bacterial cells so they can survive extreme, harsh conditions. Fungi reproduce both sexually and asexually through the formation of spores. Asexual spores are formed by the hyphae of one organism. The organisms that form from these spores are identical to their parents. Sexual spores result from the fusion of nuclei from two strains of the same species of fungus. Organisms from sexual spores derive characteristics from each parent.
What is the ladder of nature?
Aristotle, 384 to 322 b. CE, tried to use logic to build a diagram of living things. But he realized that living things could not be so easily apportioned. Instead, he constructed a ladder of nature or scale of perfection, which began with humans at one end and proceeded through animals and plants to minerals at the other end. Aristotle ranked these groups on a scale based on the four classical elements: fire, water, earth, and air.
What are the uses of chromatography?
Chromatography is used to separate and identify the chemicals in a mixture. It is useful to 1. Separate and identify the chemicals in a mixture. 2. Check the purity of a chemical product. 3. Identify impurities in a product. And 4. Purify a chemical product, on a laboratory or industrial scale.
What is the role of yeast in beer production?
Beer is made by fermenting water, malt, sugar, hops, yeast, species Saccharomyces spp, salt, and citric acid. Each ingredient has a specific role in the creation of beer. Malt is produced from a grain, usually barley, that has sprouted, been dried in a kiln, and ground into a powder. Malt gives beer its characteristic body and flavor. Hops is made from the fruit that grows on the herb Humulus lupulus, a member of the mulberry family. The fruit is picked when ripe and is then dried. This ingredient gives beer a slightly bitter flavor. Yeast is used for the fermentation process. Making beer is a complex process. One method begins by mixing and mashing malted barley with a cooked cereal grain such as corn. This mixture, called wort, is filtered before hops is added to it. The wort is then heated until it is completely soluble. The hops is removed, and after the mixture has cooled, yeast is added. The beer ferments for 8 to 11 days at temperatures that range between 50 degrees and 70 degrees Fahrenheit, 10 degrees and 21 degrees Celsius. The beer is then stored and kept at a state that is close to freezing. During the next few months, the liquid takes on its final character before carbon dioxide is added for effervescence. The beer is then refrigerated, filtered, and pasteurized in preparation for bottling or canning.
How does external fertilization differ from internal fertilization?
External fertilization is common among aquatic animals including fishes, amphibians, and aquatic invertebrates. Following an elaborate ritual of mating behavior to synchronize the release of eggs and sperm. Both males and females deposit their gametes in the water at approximately the same time in close proximity to each other. The water protects the sperm and eggs from drying out. Fertilization occurs when the sperm reach the eggs. Internal fertilization requires that sperm be deposited in or close to the female reproductive tract. It is most common among terrestrial animals that either lay a shelled egg, such as reptiles and birds, or when the embryo develops for a period of time within the female body.
What is unusual about the way the emperor penguin's eggs are incubated?
Each female emperor penguin, Aptenidites fursteri, lays one large egg. Initially, both sexes share in incubating the egg by carrying it on his or her feet covered with a fold of skin. After a few days of passing the egg back and forth, the female leaves to feed in the open water of the Arctic Ocean. Balancing their eggs on their feet, the male penguins shuffle about the rookery, periodically huddling together for warmth during blizzards and frigid weather. If an egg is inadvertently orphaned, a male with no egg will quickly adopt it. 2 months after the female's departure, the chick hatches. The male feeds it with a milky substance he regurgitates until the female returns, now padded with blubber. The females take over feeding the chicks with fish they have stored in their crops. The females do not return to their mate, and own offspring, however, but wander from male to male until one allows her to take his chick. It is then the male's turn to feed in open water and restore the fat layer they lost while incubating.
What is an organ system?
An organ system is a group of organs working together to perform a vital body function. Vertebrate animals have 12 major organ systems.
Which came first, DNA or RNA?
The first information molecule had to be able to reproduce itself and carry out tasks similar to those done by proteins. However, proteins, even though bigger and more complicated than DNA, can't make copies of themselves without the help of DNA and RNA. Therefore, RNA was the likely candidate for first information molecule because scientists have found that RNA, unlike DNA, can replicate and then self-edit.
What was the distribution of radioactive fallout after the 1986 Chernobyl accident?
On April 25 to 26, 1986, the world's worst nuclear power accident occurred at Chernobyl in the former USSR, now Ukraine. while scientists were testing one of the 4 reactors at the Chernobyl nuclear power plant. Located 80 miles 129 kilometers north of Kiev, an unusual chain reaction occurred in the reactor. This subsequently led to explosions and a fireball that blew the heavy steel and concrete lid of the reactor. Radioactive fallout, containing the isotope cesium-137, and nuclear contamination covered an enormous area, including Belarusia, Latvia, Lithuania, the central portion of what was then the Soviet Union. The Scandinavian countries, the Ukraine, Poland, Austria, Czechoslovakia, Germany, Switzerland, Northern Italy, Eastern France, Romania, Bulgaria, Greece, Yugoslavia, the Netherlands, and the United Kingdom. The fallout, extremely uneven because of the shifting wind patterns, extended 1,200 to 1,300 miles, 1,930 to 2,090 kilometers, from the point of the accident. The accident led to the release of roughly 5% of the reactor fuel, or 7 tons of fuel containing 50 to 100 million curies. Estimates of the effects of this fallout range from 28,000 to 100,000 deaths from cancer and genetic defects within the subsequent 50 years. In particular, livestock in high rainfall areas received lethal dosages of radiation.
Why don't all types of cancers respond the same way to anti-cancer drugs?
The purpose of most anti-cancer drugs is to target the overproduction of malignant cells. Different types of cancers arise from different types of tissue. Since every tissue is made up of cells specialized for a certain function, it is not surprising that different forms of cancers will have different responses to the same drug. For example, a drug that targets the overproduction of liver cells, which are adapted to filtering and monitoring the blood supply, might have little effect on nerve cells that specialize in carrying messages.
How did the discovery of bacteria impact the theory of spontaneous generation?
The theory of spontaneous generation proposes that life can arise spontaneously from non-living matter. One of the first scientists to challenge the theory of spontaneous generation was the Italian physician Francesco Redi, 1626 to 1698. In 1668 Redi performed an experiment to show that meat placed in covered containers, either glass-covered or gauze-covered, remained free of maggots, while meat left in an uncovered container eventually became infested with maggots from flies laying their eggs on the meat. After the discovery of microorganisms by Antony van Leeuwenhoek, the controversy surrounding spontaneous generation was renewed, as it had been assumed that food became spoiled by organisms arising spontaneously within food. In 1776 Lazzaro Spallanzani, 1729 to 1799, showed that no growth occurred in flasks that were boiled after sealing. The controversy over the theory of spontaneous generation was finally solved in 1861 by Louis Pasteur, 1822 to 1895. He showed that the microorganisms found in spoiled food were similar to those found in the air. He concluded that the microorganisms that caused food to spoil were from the air and did not spontaneously arise.
What are the steps of the scientific method?
Research scientists follow these steps. 1. State a hypothesis. 2. Design an experiment to prove the hypothesis. 3. Assemble the materials and set up the experiment. 4. Do the experiment and collect data. 5. Analyze the data using quantitative methods. 6. Draw conclusions. 7. Write up and publish the results.